Hello viewers, this class we are going to discuss the flower, the feature of angiosperms, reproductive feature of angiosperms. Flower is an evolutionary marvel in the sense each part of the flower is having a specific function. This can be linked to evolution as we will be discussing in subsequent parts. So, a flower is a condensed stem with a definite growth. It grows for specific length, where the different appendages are sepals, petals, stamens and carpels. As it is seen, the internode is almost reduced and the appendages appear to arise from a very common point. So, that is why it is said the flower is a condensed shoot meant for reproduction. The stalk of the flower is called pedicel, on which at the tip of which all the floral appendages are borne. Usually, the tip of the pedicel is swollen and called receptacle, to which all the appendages are attached. It is seen all plants have typically in their flowers have 4 whorls: calyx, corolla, androecium and gynoecium. Calyx consists of sepals, corolla consists of petals, androecium consists of stamens and gynoecium consists of carpel or many carpels. So, those flowers having all these 4 whorls are said to be complete. As we have seen in our practical classes, hibiscus, the typical flower, dicotyledonous flower. You see, it has a typical stalk called pedicel, and at the tip of which all the appendages are borne, but the receptacle is not seen here. So, the first whorl is calyx, then corolla, then androecium, then gynoecium. As it consists of all 4 whorls, it is called complete. And further, the 2 essential whorls of any flower is your androecium and gynoecium, as they do take part in reproduction. So, any flower having the essential whorls androecium and gynoecium is called perfect. So, this flower having all 4 whorls is called complete, and so also it is a perfect flower, and it is a perfect pentamerous flower typical of all the dicots. This one, it is lilium, this is a lily, and you can see this is a trimerous flower. Hopefully, it is well visible, it is having a good receptacle, fleshy receptacle, and I have removed some of the tepals from this, so that you can see the point from which all the tepals arise. So, now instead of 2 whorls, calyx and corolla, this has 1 whorl, that is called perianth. Wherever sepals and petals are indistinguishable, the whorl surrounding the stamens and carpels is called perianth, and the individual members of the perianth are called tepals. So, now it is also a perfect flower having stamens and carpels. Incomplete flower, we are acquainted with this plant cucurbita. Cucurbita has both monocot and dicot flowers, male and female flowers born in the same plant. So, male flowers are there, female flowers are, male flower has 3 whorls, calyx, corolla, androecium, female has calyx, corolla and gynoecium. So, both are incomplete flowers, and further I want to exemplify that or explain that this condition is called monoecious condition. As in the same house, in the same plant, both male and female flowers are born, and further these, as seen from these, they have 5 sepals as well as 5 petals. So, these flowers are also pentamerous in nature. Next, this one, the red flower, is clearly a tetramerous flower. This belongs to Ixora. Katharangani. This is perfect tetramerous flower, symmetrical flower. In this you can see all 4 petals and anthers attached to the petals. So, epipetallous condition you can find, and in the right side you can see this is the flower of Raphanus sativus, mula, radish. Very few might have seen the flower of this, but this is a cross shaped flower. So, Raphanus sativus, a tetramerous flower. This Raphanus sativus belongs to cruciform, as the 4 petals are arranged in a cross shaped manner, cruciform manner, Jesus Christ cruciform manner. So, that is why this belongs to family Cruciferae. What I mean to say, both pentamerous flowers and these tetramerous flowers are found in dicots, and trimerous flower is found in monocots. Now, I want to discuss the different whorls. What the first whorl is calyx. Calyx consists of sepals, and the sepals form the lowermost whorl, or lowermost, they are born at the lowest level. So, this flower, I think you are acquainted with this. This is datura, in Odia it is dudura. These are the leaves, but this is a flower bud. Are you able to see any of other appendages? No. What is seen? This one, only the sepal. It is a gamosepalous condition. The sepals are fused, forming a tube like structure, but excepting the sepals, nothing inside are seen. So, sepals are the outermost, they are present in the lowermost region of all the appendages, and they constitute the calyx, but their function is protection. They protect rest of the whorls from drying, and they provide an internal environment where the humidity is high. So, other floral organs develop inside this cup shaped structure of the calyx. So, now sepals are the thickest, they are the green structure of the flower, and they are the toughest, and epidermis having cuticle is also present there. That is why it is called, it is waxiest of all the floral parts, but you will find in rest of the whorls you will not find any waxy coating or any tough structure. So, these sepals are green, toughest, and they provide protection to rest of the whorl, and usually green, but in some of the flowers you will find sepals are often petal like. So, that is why sepals are called petaloid sepals. You might have seen this is the leafy nature, this is cucurbita, the sepals are fully leaf like. You will be confusing whether they are sepals or they are leaf. These are sepals, but fully leaf like, and the petals even is green. They in due course of time they will turn yellowish and become the corolla. Okay. And this is a female flower, and this one, as I told you, in certain flowers the sepals become petal like, they are petaloid sepals. The main reason is attraction, to attract the pollinators. So, this is salvia, this belongs to the family Lamiaceae, तुलसी फैमिली गछटे एथिरे सेपाल्स गुड़ा मध्य पेटाल बढ़िया होइछि दूररु सेपाल पेटाल कोटा जानि हेबो नाही किंतु दे आर इफ यू सी फ्रॉम द क्लोज विसिनिटी यू कैन सी व्हिच आर द सेपाल्स एंड व्हिच आर द petals. So, gamosepalous and gamopetalous condition is found in this, and you can see a honeybee is collecting pollens from that, but by that unknowingly it is doing the job of pollination. On the receptacle, petals are just above the sepals, which together constitute the corolla. In flower, we will be discussing more about these petals, because petals are variously colored. The color of the petals is other than green, they may be red, they may be bluish, they may be of different colors. So, the color of the petals is because of either carotenoids or anthocyanins of different color, and we will see morning glory, Ophragita, Petunia, they have anthocyanin in them, and anthocyanin is housed, that pigment is a water soluble pigment present in the vacuoles of the petals. Now, so next is your, you can see, you can do a very good experiment at home whether the petals contain carotenoids or anthocyanin. I have taken hot water in a glass and I put the flowers therein, and the other one is cold one. When in the hot water it is put for 2 to 3 minutes, then definitely the pigment comes out, it is leaching out, as it is water soluble housed in the vacuole. When we put it in hot water, the tonoplast, that is vacuolar membrane, plasma membrane, they are disrupted and the pigments they come out. Whereas, in the other one, when it is put in cold water, normal temperature at 25 degree centigrade or so, it does not, as the cells remain intact. But if you put this marigold, Gendophylla, in the same way, you will not get in hot water, the pigment will not come out. So, it signifies if it is not coming out in hot water, it is not anthocyanin, rather it is your carotenoids. So, now about the petals are important in attracting pollinators. Petals have different sizes, shapes and color and arrangement. So, now generally if you look at the nature, some flowers bloom in night. Night blooming flowers are usually white in color, they are white, they lack any pigment, as it is found in Tagoro, Tabernamontana, and in some other plants. But other, you see this one, I have written this is a day blooming flower, this name is morning glory, the name itself signifies it blooms in the morning, and this is a color plant. So, color of the flower is enough to be visible to the insects and pollinators, but white flowers attract the pollinators by their fragrance, but these flowers attract mostly by their color. Now, if you look at this, this is a flower or inflorescence of Asteraceae family. Now, you can see one yellow patch, and this pattern is used by, or the flower use this pattern for attract a specific kind of pollinators. You can see Apis dorsata is pollinating or collecting pollens from this flower. Next, this one very interesting one, this is Myrtania, and in Odia it is called Baghonokhi, and this is the flower, it is bilaterally symmetrically flower, and a specific kind of spot is present at the mouth of the Gamopetalous corolla, and that is called honey guide. That honey guide is used, or plants use this for attracting certain kinds of insects. And further, everybody wants to advertise himself. Day blooming flowers with huge big flowers and bright color, they attract pollinators, but those who are wind pollinating, those which are pollinated by wind, these plants never invest in formation of petals. Here you can see rice, rice does not produce sepals and petals, rather highly reduced perianth called lodicule is formed therein. So, like rice, other wind pollinating plants do not invest energy and biomass in producing the perianth corolla. Other flowers, day blooming flowers attract pollinators, but wind pollinated flowers has nothing to attract because wind blows. So, that is why these plants have evolved highly reduced perianth. So, the perianth is represented by lodocule here, and other adaptations of these wind pollinator flowers, as you can see, they have feathery stigma and the anthers, beautiful anthers, 6 in number in each flower, right. They are highly swinging in the air and they shed their pollens, so that they can be collected by these feathery stigmas. So, now next two essential whorls are the stamens and carpels, and the stamens collectively called as androecium. The number of stamens vary from flower to flower. As we have seen in our hibiscus, we write the number of stamens is infinity, large number of stamens are there. So, the stamens are usually called microsporophylls, as they produce microspores in their spore producing structure called anther. Generally, a stamen is referred to as microsporophyll and it is the male part of the flower. Truly speaking, the anther is not the male part, rather it on meiosis produces microspores, which on a further development give rise to the male gametes. As male gametes produced in the anthers, that to stamens, that is why are called microsporophylls, and all stamens constitute androecium. Andros means male. Okay. So, each stamen consists of a filament, a stalk, and the real spore producing structure called anther. As revealed, a typical anther consists of two halves, first 2 equal halves, and then each half again further has 2 lobes. So, in toto a typical dicot anther or monocot anther has 4 longitudinal lobes or 4 longitudinal structures called sporangia. That is why a typical anther is said to be dithecous and tetrasporangiate type. As we see this plant is a monocot plant and it is amaryllis. I think you can see there are 6 stamens. Each stamen consists of 4 longitudinal structures, 3 are seen from this side and 1 is the other side, it is not visible. So, usually 4 longitudinal structures, 4 sporangia, they are arranged in this anther. What is this? Hopefully, all of you are acquainted with. This is taken generally with a camera, and these are the 2 essential structures of hibiscus, the stamens and the carpels, but entire carpel is not seen here, rather the 5 stigmas are shown here. 5 stigma, they are hairy stigmas, beautiful condition to enjoy, because the anthers have dehisced, they have opened apart releasing the pollens. Now, these pollens are attached to the stigma. So, carpel, the innermost whorl, gynoecium, the innermost whorl, it may consist of 1 carpel or more than 1. All legumes, all the beans, they have 1 carpel, they are monocarpellary, but if you look at lady's finger, it consists of 5 carpels, but all the 5 carpels in hibiscus, in lady's finger, all the 5 carpels are fused together, forming a compound structure called pistil. So, the flower which is having only pistil or carpel, that means female flowers are called pistillate flowers. What we say in cucurbita, we can find female flower called pistillate flower, or else it has male flower called staminate flowers. Okay. Those having both stamens and carpels as essential structures, they are called as bisexual flowers. Now, each carpel or a pistil has 3 parts. The uppermost part is the stigma, which is projected beyond the flower, and it is meant for, or it is the site to which the pollens land on, or stigma is the site to which pollens bind. So, it is pollen landing surface, and the basal part of the carpel is ovary. It is the swollen part. The basal part of the carpel is the ovary. Ovary is a bulbous structure, and the structure that joins, you can come to this point, the structure that joins the ovary to the stigma is your style. So, style, stigma, style and ovary, they have different functions assigned to them. Stigma is the landing ground of the pollens, style elevates the stigma above the petals, so that the pollinators can visit the stigma, and the basal portion is the ovary. Ovary houses one or more ovules. We have seen mango. Mango, the ovary has only one ovule in it, but if you look at a beans, if you look at a pea, Pisum sativum, it has so many ovules in it. So, the number of ovules in each ovary may vary depending on the plant type or species type. So, now the function of the carpel is to house ovules inside the ovary, and the ovary consists of, as we have seen, a ovary contains ovules. Generally in angiosperms the ovule is anatropous type. The anatropous ovule is like this, it has a funiculus, the stalk of the ovule is called funiculus, and it has the two integuments, outer coatings, and this the central mass of cells is your nucellus. This is the nucellus, and these are, this is outer integument, and this is inner integument, and the central mass of cells is the nucellus, and the integuments, the coverings, they encircle the nucellus from one side, leaving a small aperture at the top, and this is your micropyle. This is your micropyle, small opening, and the other end is called chalaza. So, this end is called micropylar end, and this one is called chalazal end. Now, 70% of the angiosperms have this kind of ovule, and this ovule is your anatropous ovule, where the ovule is curved in such a way that the micropyle is near to the funiculus. So, now this ovule is the structure of the sporophyte. It is deep seated in the ovarian cavity. This is the site where you can see here one of the cells become large and visible, okay. And this is a 2n cell, all the cells are 2n, but one of the cells of the nucleus becomes megaspore mother cell, it grows. Its fate is to go for meiosis, or this is MMC, megaspore mother cell. Now, this megaspore mother cell will further go for meiotic division, resulting in 4 megaspores. Main difference between microspores and megaspores is microspores are produced in a large number, because microspores lead to pollen grain formation, and pollens travel from flower to flower, but megaspore is sedentary, it remains attached to the parent plant, they are not lost, but pollens are lost in the transit. So, to cope with, to bear with the loss, the damage of the pollens, so plants produced large number of microspores, and so also subsequently they are turned to pollens. Now, we are going to discuss how the male and female gametes are formed, because male and female gametes are necessary for fertilization to take place. So, male gametes are produced in the male gametophyte, whereas the female gamete is produced in the female gametophyte where they are present. As we recall, the male gametophyte is a pollen grain and the female gametophyte is an embryo sac or the female gametophyte. It is present inside the ovule. So, for development of male gamete from male gametophyte, we have to discuss the anther and its anatomy and the meiosis and other aspects, so that development of male gametophyte can take place. Now, a stamen is a microsporophyle. The filament is just the subtending part. It provides nutrition to the anther, but the anther is itself the spore producing structure. We discussed anther is tetrasporangia type, and the inside the anther, the wall we will see it consists of 4 layers, and inside the anther or inner to the anther wall in the center there is a tubular structure called microsporangium or the pollen sac. So, in the pollen sac we will find initially the archisporial initials. These initials grow and they are destined to form microsporocyte. The discussion can be held in 2 points. One is microsporogenesis. Microsporogenesis is the process in which a microsporocyte present in the pollen sac or microsporangium, which is a diploid cell, it undergoes meiosis producing 4 microspores. These microspores, they are formed in a group of 4 called spore tetrad, and they are glued together and they become free from each other subsequently. And this haploid microspore further grows in size, divide mitotically, resulting in a 3-celled structure to form a pollen grain. That may be a 2-celled initially and subsequently 3-celled structure. So, a matured pollen grain is a 3-celled structure. So, in that matured pollen grain we will be having 2 sperms, male gametes. So, that is why the pollen grain is the male gametophyte, and formation of microspore from microsporocyte or microspore mother cell by way of meiosis is microsporogenesis. Once microspore is formed, then it grows in size, then mitotic division takes place, we will be discussing, resulting in a 3-celled structure, and out of which 2 are the sperm cells or sperms male gametes. So, that is why this part starting from microspore to pollen grain formation is microgametogenesis. Now, let us have a look at the anther wall. The anther wall consists of 4 layers. From outer side, the outer one is epidermis, brick shaped cells, flattened cells, right. It is a continuous structure. Inner to that is endothecium. Endothecium is a single layer cells. These are large cells, these are very large cells, and the endothecium, they are responsible ultimately for dehiscence. And inner to endothecium, epithecium is single layered, endothecium is a single layer of large cells, and middle layers, they are very small cells, 2 to 3 layer cells inner to endothecium, and the innermost layer of the wall is the tapetum. That is highly significant. Okay. So, tapetum is a layer, the cells are very big and they serve as nursing cells. Their function is to nurse or nourish the developing pollen grains. So, now besides this, let us have a look. In the pollen sac, the diploid archisporial initial cells, they transform them by means of growth to meiocytes, the cells which undergo meiosis, and these are called microspore mother cells. Okay. These microspore mother cells, each of them undergo meiosis, resulting in 4 microspores. These 4 microspores, which are haploid, they are bound to each other in a condition called spore tetrad. Now, microspores are initially glued together, as will be seen, but later they go on separating from each other, because the 4 microspores, they do not remain together, rather they become free from each other. So, definitely what is imminent? Definitely the glue, the cementing substance which hold all the 4 together, they must be, that glue must be dissolved. So, that is why they must be separated, and that is the glue is callose, a polymer, and the callose is dissolved or digested by the enzyme callase. Further, the callase enzyme is secreted by the tapetum. So, tapetum on secretion of callase, which digest the wall surrounding all 4 microspores and the cementing substance in between them. So, ultimately the microspores are unglued, separate from each other, and once the microspores are free from each other, the term pollen grain is used. So, ultimately the immediate product of meiosis, they are microspores. Once the microspores, they are free and have independence, the term pollen grain is used. So, pollen grain, young pollen grain, and subsequently they mature to form matured pollen grain. Now, the function of your tapetum is very important. By way they undergo death, the tapetal cells undergo a kind of death called programmed cell death, as a result of which they secrete not only enzyme, rather carotenoids, lipids, and many substances which are necessary for the development of the pollen grain. So, let us look at this. This is just a microsporangium. Now, the microsporangium has so many microspore tetrads. In the right, you can see 4 microspores or young pollens, they are released from a tetrad. So, this action is done by a enzyme called callase. So, now each microspore grows in its size and becomes a pollen grain. Important is the pollen develops or microspore as it grows in size by absorbing liquid substances from the pollen chamber or microsporangial chamber, then they increase in size, then they become vacuolated, and ultimately by that time they develop 2 kinds of wall investment. So, the pollen develop wall around it, the inner wall is intine and outer to that is your exine. Now, what is seen exactly? The pollen grain as it grows in size, it develops wall around it. So, the inner wall is intine and outer one is exine, and there is a haploid nucleus here, and this haploid nucleus again undergo asymmetric mitotic division, asymmetric mitotic division. Subsequently, this divides resulting in 2 nuclei, and one nucleus is here, and this is your generative cell and the large one is the vegetative cell. So, this way initially the growth takes place and wall investment goes on around it. Now, this is a two-celled pollen grain. This is a two-celled pollen grain. Now, a very small cell attached to the cell wall, that is the generative cell. As the name suggests, why we must understand why it is so called. The generative cell on further mitosis, it undergoes a mitosis and results in two sperm cells. So, that is why the name generative cell. Vegetative cell, as the name suggests, this is the pollen tube cell, because this will give rise to pollen tube while it lands on the stigmatic surface. Now, thing is, this is a generative cell. This cell becomes free from the cell wall, and ultimately a cell which is suspended, it is a spindle shaped cell, it is suspended in the vegetative cell cytoplasm. Why this becomes spindle shape? It is assumed that if it is a spherical cell, then it may face difficulty while passing through the pollen tube, but if it becomes spindle shape, then it is easy. So, that is why this generative cell now again undergoes another mitotic division to form two sperm cells. Ultimately, here two sperms are produced here. These sperms are haploid sperms and mostly filled with nuclei and little bit of cytoplasm with organelles. Usually, the sperms do not have plastids in them, right. What about this exine? Exine is the outer coating, and exine is formed of a substance called sporopollenin. Exine is made of sporopollenin. This is of very much significance. It is a polymer, or it is a compound formed of carotenoid derivatives. So, this is deposited. Two things we must remember: the inner wall is intine, the intine is formed of callose, a soft substance, it is not hard substance. So, the callose wall is deposited by the microsporoprotoplast itself, whereas the outer investment, the outer wall, the exine is deposited by the tapetum. Along with sporopollenin, other lipid-rich substances are also deposited around this, forming pollen kits. Now, question is, sporopollenin is one of the toughest material in the living world. They are bioresistant, they cannot be degraded by acid or alkali. Therefore, pollen grains with sporopollenin in its coat are long lived, they are not easily destroyed. Pollens are resistant, as they are resistant to degradation, they are well preserved in the fossil deposits, in the coal deposits. That is why the pollens are suitable for fossil study. The branch of plant biology that deals with pollens and linked to biosystematics is your palynology. And here you can see the outer coating exine is not deposited all around. In certain regions, the sporopollenin is thinly deposited or absent. So, the area that may be linear or circular, the area where the sporopollenin is not deposited, these are said to be germ pores. The sites in future a germ tube, pollen tube will emerge. You can see the exine, because of sporopollenin, is variously sculptured. It has, the outer coating has ridges, furrows, lines and so many things. You see this. This is transverse section of anther of datura. Easily you can see the pollens in the first left diagram, the pollens are already released, only the anther wall is left, but the pollens were therein, there is no dehiscence, but when we took the transverse section, then pollens are released from that, but naturally it has not dehisced, the anther has not dehisced, not burst. So, therefore, 4 pollen chambers are now seen, and the second one you can see the pollens. These are the pollens of hibiscus rosa sinensis mandar. In light microscope we have taken this, and you can see the pollens have spine like structures in their exine, they are yellow in color. If you look this and other pollens, they have different patterns in their exine. Therefore, the pollen shape, the exine pattern is species specific. Those who are specializing pollens, palynologists, easily they can tell of what plant this pollen is or what family this pollen is. So, pollens are a clue to identification of plants. So, now pollens are well preserved in fossil deposits, as I told you. Pollens cause allergies. This parthenium, which generally called carrot grass, because the inflorescence of this looks like carrot inflorescence. So, that is why it is called carrot grass. Sometimes it is also called congress grass, because these seeds came to India when in 1950s wheat was imported in congress regime. For that reason probably it is called congress grass. Okay. So, the pollens, this is a weed, parthenium is a weed. This grows elsewhere in the rail lines and elsewhere. So, their pollens cause allergies. Some people they sneeze often because of pollen, because pollens are allergic to some people. And further, as pollens are very small structures, they are released in large number from the plants, and they are rich in lipids and other valuable substances, minerals in them. Therefore, pollens are in form of tablets and syrups are used by athletes for energy purpose. They take this. The haploid nucleus of the microspore. Now, I can say here how exactly this male gametophyte developed. Male gametophyte developed. Now, this haploid nucleus of the microspore moved to one side, there it divided, but mitotic division, nuclear division is symmetric, but cytoplasmic division remained asymmetric. Around this there was very little cytoplasm, then only it resulted in one generative cell and one vegetative cell. The vegetative cell nucleus is somewhat convoluted, it has more surface area and it controls the entire vegetative cell. Okay. Now, the generative cell, as the name suggests, it will produce it produces the sperm by meiosis, by mitosis. It may be taken note, prior to division of the generative cell to 2 sperm cells by means of mitosis, there once it undergoes DNA replication. So, generative cell nucleus undergoes DNA replication before the cell divides. Now, finally, 2 celled pollens and 3 celled pollens we can get. A 2 celled pollen can be released from certain plants. 75% of the angiosperms they shed their pollens at 2 celled stage, which is a immature pollen grain, and these 2 celled pollens, when they land on their right kind of stigma, there or while sending the pollen tube, there the generative cell divides, resulting in 2 sperm cells, completing the process of formation of sperms. So, there the male gametophyte becomes 3 celled, and in others, while it is in the anther, the pollen development becomes complete and it becomes 3 celled. So, it does not matter whether it 2 celled or 3 celled, ultimately on landing 2 sperms are there in the male gametophyte. Okay. So, you can see this is the sculpturing of pollen exine, this is scanning electron micrograph of a pollen from Caeta. This plant we do not know, but this is a these are the pollens of this Caeta plant, and you can see the different patterns of deposition of exine in it, sporopollen in it, and the germ pores are clearly visible. Now, the 2 sperm cells are not surrounded by heavy thick wall, as sperms ultimately will meet the egg for fertilization. If a heavy wall would be invested around, definitely they are not going to fuse. So, that is why sperm cells have thick, very thin wall, that is either callose type cell wall, and the 2 sperms they are suspended in the cytoplasm of the vegetative cell. Now, once the pollens are ready, the anther wall dehisces in a particular line, resulting in release of pollens. So, you can see this in this lilium, the anthers are split longitudinally, resulting in release of the pollens. So, this is the summary how the microsporogenesis and microgametogenesis results in 2 celled pollen grain. Then megasporogenesis. Megasporogenesis, that is formation of megaspore. Already we have discussed ovary houses. You see the structures, carpel consists of stigma, style, ovary. Ovary is the bulbous structure, it has wall and it has placenta in the center. So, to the placenta ovules are attached by means of funiculus, and each ovule is surrounded by integuments, and one opening is left at the apex, that is your micropyle, and in the center there is a megasporocyte, that is megaspore mother cell. You can see this. This is the transverse section of hibiscus ovary. You can see the at the center pentaradiate placenta is there, and to each placenta there is attachment of the ovules. Now, within the nucellus, the megaspore one mother cell undergoes meiosis, resulting in 4 linear structures, that is called linear tetrad. But only one of these 4 megaspores will survive and rest 3 will degenerate. Out of 4, the 3 micropylar ends, they undergo programmed cell death or they degenerate, resulting in only 1 megaspore left at the chala end. So, this is about megasporogenesis. Only 1 megaspore survives, which is a haploid structure. Further, this megaspore increases at the cost of the dying cells. The nucleus divides thrice, 3 successive divisions of mitotic divisions of this megaspore nucleus takes place, resulting in 4 or 8 nuclei, 4 at each end. Let us have a discussion like this. This is the initial megaspore, this is its nucleus. This nucleus divides by 3 successive divisions, mitotic division, resulting in first when 2 nuclei are formed, here 1 nucleus, here another nucleus. Likewise, this again divide twice, this again divide twice, resulting in n 4 nuclei at each pole, and this cell is vacuolated. Now, redistribution of the nuclei takes place, 3 remain at this end, 3 remain at this end, and 2 from opposite poles they migrate to the center. Now, ultimately following this there is free cell formation. So, you can look at this, when there is redistribution of the 8 nuclei, 3 remains at the micropylar end, 3 at the chala end, and 2 in the center, and then surrounding each nucleus there is cell wall formation along with little bit of cytoplasm. So, this results a 7 cell structure. Those remaining at the micropylar end constitute the egg apparatus. The central one is the egg cell and the 2 side cells are the synergids, and in the center the cell is primary endosperm cell that has 2 haploid nuclei, and those at the chala chala end constitute the antipodals, and this 7 cell structure is the female gametophyte that does not leave the plant, that remains in the ovule itself. So, now as this type of female gametophyte develops from a single spore, that is monosporic embryo sac, and it was discovered in polygonum. So, that is why the monosporic embryo sac is polygonum type, typical of the angiosperms. Difference of male and female gametophyte is, male gametophyte is 3 cell structure independent of the sporophyte as it leaves the plant, and it tolerates desiccation chemicals because it leaves the parent plant, whereas the female gametophyte is a 7 cell structure dependent on the female gametophyte, remains attached to the female gametophyte and susceptible to desiccation. Now, this retention of the female gametophyte in the embryo in the ovule is necessary for seed formation. Thank you.